And it's my pleasure to introduce Kristen Brennand from the Icahn School of Medicine at Mount Sinai in New York. She's a stem cell biologist and specializing in vitro work and iPSCs as a model of schizophrenia. The floor is yours, Kristen. Thank you so much.
Thank you. I can't share my screen until Robert stops, though.
Okay.
So it is my great pleasure. I think, no.
Yeah, now you should be possible.
There we go. Share. And okay. It is my great pleasure to get to share our work with this community today from the comfort of my own kitchen. And so what I want to talk with you guys about today is the work that we've been doing to apply stem cells to explore the genetics underlying psychiatric disease. And I'll note there's tiny little icons on each slide. If there's a green camera, feel free to screen grab or tweet it. And if there's a red, please don't. So why are we using stem cells to teach us about something as complicated and holistic as psychiatry? And I think that there are two things that we're really hoping to provide to the field. And one is a new avenue by which to potentially help better diagnose patients. Genetic-based diagnosis, I think, is something that's very obvious to this group. And we really think we have a tool to help functionally validate and link these variants that you guys are uncovering to their function in a cell type specific and donor dependent manner. And I think second and bigger picture is the real goal is, of course, not just to diagnose patients, but to treat them, and to do that either after the onset of symptoms or ideally prior to the onset of symptoms. And so with genetic-based diagnosis, we can really start thinking about drugs that one might deliver to high-risk kids prior to symptom onset. And so what we're talking about here is precision medicine, whereby we consider all of the risk factors and the interactions between them and any given patient, and how those impact not just clinical outcomes, but treatment response. And this slide is nothing new to any of you, but I want you to consider how you would feel as a stem cell biologist, somebody who really didn't know the full complexity of the genetics behind this data, and somebody who wanted to functionally validate a hit. And so this is why I'm so grateful to work with fantastic geneticists like Pamela Sklar and now Eli Stahl and Laura Huckins, because as a stem cell biologist, and I'm not sure if you can see my cursor, I was of course drawn to that very, very tall one at the top of the graph. And they stopped me. They said this was a horrible locus to go after and pointed us instead at this one here, at the gene furin. And again, they really walked me through understanding why of the entire, so at the time it was the 108, but it's still true now at 145 loci. At 145 genome-wide significant loci, what happens at the furin locus is the only time it happens, and that's that you have the single top SNP for GWAS, so that's the y-axis, is also the single top SNP for brain EQTL of a coding gene. So at furin, you have that one SNP in the top right corner that is most significant for both disease and brain expression. And so that top SNP here is a putative causal SNP, and it was one that we thought we might be able to edit by CRISPR. Now, the next best examples look more like what we see at SNP91, where you have 20 or 30 SNPs clustered in the top right-hand corner. But we don't know what's going on in that top right-hand corner. We don't know if there's one putative causal SNP in there, or if each of these 30 SNPs are contributing 3% of the risk at the locus. And so what I'll talk to you about today is the stem cell and CRISPR-based strategies by which to interrogate risk at locuses that look like, or loci that look like furin or SNP91. And so the furin work was led by a very talented former postdoc in my lab. I'll forever call her the bravest postdoc in my lab. I showed you on the previous slide the best data for the furin locus, but now what I'm showing you is the other side of the same data. So this is the expression in the Common Mind Consortium brains by genotype at RS4702, the furin SNP linked to schizophrenia. And what you can see is, yes, it is quite significant because there's 600 brains, but the error bars there are daunting, right? And the reason these error bars are so big is, of course, each of these brains came from a different individual. And each of these individuals varied at many locations beyond just the furin SNP. I see some questions here. I'll leave those till the end. So each of these donor brains varied at many genotypes genome-wide. They came from donors, some of whom had schizophrenia, some of whom did not, who had different antipsychotic treatments at the time of death, different drug and alcohol abuse histories. They died at different ages, of different causes, and had different RIN values. And so our hypothesis was that if we could do this as a controlled experiment in the lab, whereby nothing was different except for AA to GG genotype, our hope was that the effect sizes would be the same, but the standard deviations would be much smaller. And so that is what Nadine undertook to test. Now, this was a very hard edit to do. She started this back in like 2014, I think. And the reason it was hard is we couldn't use any of the standard tricks that were developed at the time to increase the efficiency of CRISPR editing. And that's because this was a single non-coding SNP, and editing a second single non-coding SNP downstream in the PAM was just going to confound the analysis. But she found a way, and she got these edits. And when she did, she was able to show that in isogenic neurons from the same donor cultured in the same plate side by side, when nothing more was different except AA to GG genotype, you could actually see a significant change in expression of furin levels. Moreover, in the time that it took her to make these edits, it was discovered that this SNP in the 3' UTR of furin is also in the binding site of a microRNA, miR338. And if you inhibit miR338 in those same neurons, now you can completely eliminate this EQTL effect. So it's a cell type and context-dependent EQTL in neurons. She was further able to show that doing nothing more than changing genotype of a single non-coding SNP was enough to manipulate neurite length and neuronal activity. We've been moving forward asking questions now about background. So these first edits were all done in the control background of average polygenic risk. And this is now unpublished data led by a new postdoc in the lab, Christina Rebick. And we're asking, to what extent do these SNP effects vary if you do the same edits in controls with particularly low or particularly high polygenic risk for schizophrenia? So the same edit in both backgrounds. These are in a repository of iPSC, about 1,200 controls from the SERM collection, and Kevin Egg and Steve McCarroll helped us find these extreme iPSCs. And this is just our first pass, the very first pass. She's got edits now in low, average, and high PRS donors. This is that same activity assay. And it looks like we're more sensitively resolving AA to GG genotype changes in those high PRS donors. Now, there's a lot more work to be done here, but I think it's a really exciting avenue to consider that we can now really tease out cell type and donor-specific genotypes effects. Now, and beyond that, there are so many more genes to look at than just furin. And if you're not able to find a single putative causal SNP to edit, you need another strategy. And so here for the next genes at the top of the list, we're using CRISPR activation or inhibition to manipulate from the endogenous promoter the expression of these top genes. And we can do it in the disease-relevant direction by taking into account the EQTL direction. So here I'm just showing you by RNA-seq that whether we do CRISPR-A or CRISPR-I for two of these top genes, SNAP91 and tSNARE, the top gene that's changing is the target gene, SNAP91 or tSNARE, but a few other genes are also significantly changing. And those other downstream genes that are changing seem to be enriched for brain genes, particularly synaptic genes. We can see changes in synaptic puncti number by up and down regulating SNAP91 and tSNARE. And finally, reciprocal changes in SNAP91 levels up in the top panel and down in the bottom lead to reciprocal changes in synaptic activity by electrophysiology up in the top and down in the bottom. Now, of course, common variants don't change in isolation. The really interesting question is what happens when you change them together. And so here, what Nadine and Sukhmanho did together is they did individual and combinatorial perturbations of these SEAM4 genes in the disease-relevant direction. So up for SNAP91, tSNARE, and CLCN3, and down for furin. We have RNA-seq of these individual perturbations. You can computationally predict what you think should happen when you change them all together. And then you can actually do a combinatorial perturbation side by side on the same plate and ask how well the model predicted the outcomes. And so if the model is exactly right, we call that no synergy, but sometimes genes were too much up or too much down relative to predictions. And so in fact, about 82% of the time of genome-wide analysis, genes changed exactly as the expected additive model predicted. but 7% of the time genes were too much down and 11% of the time they were too much up. What types of genes were were down relative to expectations? Well, they were actually synaptic genes, and all of the major neurotransmitter classes were captured here. And those that were too much up, well, they're enriched for disorder genes, particularly the rare and the common variants associated with bipolar and schizophrenia. So what we're really saying here, I think, is that environment context matters. It's important to look at these genes in the combination of the others. And here we're just looking at 4 common variant genes, but the next experiments moving onward are 20 genes, ideally 100, 200 of these genes. We really have to understand the interactions between these genes to get a handle on common variant effects. Now, of course, everything I've talked to you about now are based on the proximal targets, those genes close to the SNPs. But the DNA is not packed into the cell in a straight line. We know it's compacted, and it's compacted in a very deliberate and organized cell type specific fashion. And these loops can bring those schizophrenia risk variants close together to genes that are much more distant in the linear space. So this was work led by Prashant Rajarian in collaboration with Sharon McBarian, where he applied high-C 3D mapping to consider cell type specific interactions in stem cell derived neurons, glia, and neuroprogenitor cells. And so if there were 224 genes close to the 145 schizophrenia loci at the time, he added a few hundred more by looking at cell type specific high-C. He looked at RNA and protein interactions in this group and saw enrichments, but he also functionally validated that these long-range interactions were manipulating expression of schizophrenia risk genes. So here there was a loci where there was 4 schizophrenia GWAS SNPs about 100 kilobases away from the protocoherent alpha locus. He used CRISPR to delete those SNPs and was able to show that deleting these risk SNPs changed expression of 2 of the 3 genes at the loci in the disease relevant direction. Finally, with the last few minutes that I have, I want to change gears and talk about rare variants. So these are those highly penetrant rare mutations, in this case deletions, that are very likely to result in disorder. I'll talk to you about some work on orexin-1. So it's the second most significant schizophrenia rare deletion, the most significant for autism, the only one of the ones on this slide that impacts a single gene, but not to make it simple, this deletion is non-recurrent. It varies between cases, the boundaries, and orexin-1 is predicted to be spliced in hundreds of different ways. So this is a small cohort because they're rare variants collected in collaboration with Judith Rappaport. The study was led by Aaron Flaherty in collaboration with Shuja Zhu, a former postdoc in my collaborator Gong Fang's lab. So one of the first things Aaron was able to show is that patients from neurons from patients had reduced neurite outgrowth. So here I'm showing you 4 patients. The 2 in blue have 5' deletions where the promoter and first 2 exons are missing, and the 2 in red are 3' deletions, missing the second, third, and fourth from last exons. The blue ones are a mother-son pair. The red ones are monozygotic twins. And again, when you look at neuronal activity, using the same assay I mentioned earlier for the furin, you see reduced activity in patient-derived neurons. So this is a time course where you're monitoring population-wide activity. The controls go up with time and the patients really don't. This is a standard differentiation with a mixture of glutamatergic and GABAergic neurons, but if you do it through a different method that yields entirely glutamatergic neurons, you see the same effect. So patient cells don't fire as much. The question being why? They fire less because one critical orexin-1 isoform is missing, because all the isoforms are decreased by 50%, or because there's new mutant isoforms not seen in controls. We added long-range and short sequencing of the orexin-1 locus to ask this question. We were able to identify 31 unique mutant isoforms from the 3' cases not found in the controls. And in purple, we're indicating abundant. So you can see that these mutant isoforms are some of the most abundant in the patient neurons. We found evidence that 49 or 50% of the wild type isoforms were significantly decreased in patient neurons. Another 28 wild type isoforms were not even detected in the patient neurons. But again, these are some of the least abundant in the controls. And so overall, there's a mass dysregulation of orexin-1 splicing repertoires. I'll add that the mutant isoforms were not found in postmortem brain, and most of the controls were, especially the most abundant ones. Finally, Aaron did some functional studies to look at the impact of overexpressing these isoforms in isolation. So if this is control neuronal activity on the left, adding a single wild type isoform does not change activity, but adding one of any 4 mutant orexin 1 isoforms dramatically reduces neuronal activity. So control neurons are impaired by a mutant isoform expression. Those 5' patients who are not predicted to have mutant isoforms have reduced activity that is rescued by overexpression of any one of 4 different wild type isoforms. Whereas the 3' patients, the ones who do have mutant isoforms, we can't seem to make them better, whether we overexpress wild type isoforms, and we can't make them worse if we overexpress mutant ones. And so overall, we think phenotypes are happening through 2 independent mechanisms, both the loss of orexin 1 dose and the additive effect of mutant isoform activity. I think this might be more broadly applicable because if you go back into the PGC rare variant data set, there are 35 orexin 1 cases that have been genotyped, and we think about 1 in 5 of them might be making mutant isoforms. And so the future studies then would be to synthesize these predicted mutant isoforms in these other cases and ask what effect they have. So overall, what we're really envisioning here with stem cell models is bridging the gap from genotype to function. And so I think stem cells and CRISPR are really good at understanding the link between DNA and RNA levels and the links between RNA levels and synaptic function. The ultimate goal of doing drug screens either at the level of gene expression or synaptic function, such that we can predict patient treatment options. And with that, this is my final and very most important slide. These are all the people who would normally be in a lab hard at work getting stem cells and instead are trapped in small apartments in Manhattan and very eager to hopefully one day get back in the lab. But without them, there really would be no data to share with any of you today. And I'm so grateful for each of them for their hard work. Today's talk was led by work done by Nadine, Aaron, Prashant, and Suk in collaboration with Gabriel, Gong, and Sharam. Thank you.
Thank you, Kirsten, so much. This was amazing. We've got one question in the Q&A for you, and it's from Huanwai Wang. Which region of the gene furin, like exon, intron, or promoter, is the SNP rs4720 located in?
Sorry for not making that more clear. It's in the 3' UTR, which is why the microRNA binding site seemed more relevant there.